Yes, students, welcome back to the part 2 of biotechnology principle and processes. In our previous class, we have seen that using the restriction enzyme, the piece of DNA can be cut into further pieces at our desired location. Now, the treatment of DNA with restriction endonuclease produces a number of DNA fragments of variable length. They are separated through gel electrophoresis. Gel electrophoresis is a technique of separating biomolecules under an electric field when placed over a matrix of gel. Cathode and anode are present at two ends of the instrument. The matrix is generally made up of gellidium based agar polymer called agarose. The DNA fragments are placed over cathode end. The electric field is activated. The DNA fragments are negatively charged. They move towards the positively charged anode. Agarose has a sieving effect. Larger fragments move slowly, while the smaller fragments move fast and reach the farther end. Now, DNA fragments cannot be seen in visible light and without staining. They are stained with ethidium bromide and made visible through UV radiation as bright orange colored bands. The bands are cut out of agarose gel and extracted. This step is called elution, or they can be absorbed onto a nitrocellulose membrane by the process of Southern blotting. The purified DNA fragments are then used in constructing recombinant DNA by attaching them to cloning vectors. The DNA library or gene library can be constructed by this method, and the genes are identified through their products. Now, as I said, we need vectors to clone the DNA. The vectors are the carriers of the vehicles of desired DNA fragment, which can undergo independent replication to increase copies of desired genes. Example: plasmids, bacteriophages, etc. Now, a cloning vector must have its own origin of replication or ORI, selectable marker for identifying recombinants from non-recombinants, and preferably a single recognition site for cloning. There are 4 types of cloning vectors: plasmids, viruses, cosmids, and artificial chromosomes. Plasmid vector for plants are different from that of the others. Plasmids. Plasmids, they are the small extranuclear circular DNA which carry extrachromosomal genes. in case of bacteria and some fungi. They replicate independently. The best known vectors which are available commercially are PBR 322 and PUC 18. The PBR 322 is the plasmid discovered by Bolivar and Rodriguez, who designated it as 322. PUC series of plasmid vectors are named so because they were initially developed at the University of California. They all have a specific origin of replication. Origin of replication is a DNA sequence which is specialized to initiate replication. The sequence also possesses nearly replication control, which determines the number of copies it would form. Therefore, the selected plasmid should have an origin of replication that supports higher copy number. Now, selectable marker. The selectable marker are the reporter genes that contains a trait suitable for artificial selection. A plasmid has a selectable marker. Normally, genes encoding for antibiotic resistance like ampicillin, chloramphenicol, tetracycline, kamamycin are considered useful as selectable markers. For E. coli, PBR 322 has two antibiotic resistance genes, ampicillin resistance and tetracycline resistance, while PUC 18 has only ampicillin resistance gene. Selectable marker is meant for distinguishing a recombinant from non-recombinant DNA. It also helps in eliminating the non-transformed bacterial cells from the transformed bacterial cell. Three, cloning or unique recognition sites. Recognition sites, a site where alien DNA can be inserted, should preferably be one and responsive to commonly used restriction enzyme. Occurrence of more than one recognition site will cause DNA fragmentation and disturb gene cloning. In PBR 322, the alien DNA is ligated generally in the area of BAM-H1 site of tetracycline resistance gene. The recombinant plasmid does not possess the tetracycline resistance but continues to have ampicillin resistance. The plasmids are passed into bacteria during transformation. The non-recombinants can flourish in medium having both ampicillin and tetracycline, whereas recombinants can grow only in ampicillin containing medium but will die out in tetracycline containing medium. Now, see students, this is a very cumbersome procedure requiring antibiotic containing plates. Therefore, alternate selectable markers have been produced. They use chromogenic substance or XGAL. The common recognition or cloning site is lac gene Z that codes for the enzyme galactosidase. Insertion of alien DNA into it inactivates the DNA and prevents the synthesis of the enzyme galactosidase. The chromogenic substances produces pale blue colonies. If the plasmids do not have inserts and white colored colonies of the recombinant plasmids are present. Now, as I said, the cloning vectors for plants and animals are different from that of the bacteria and bacteriophages. The tumor-inducing plasmid TI of the bacterium Agrobacterium tumefaciens has been made non-virulent by deleting the part which is responsible for its virulence. However, the modified plasmid does not contain the tumor-inducing gene or the tDNA. It is, however, employed as recognition or cloning site. A foreign gene is inserted in tDNA region of the plasmid and is allowed to infect the plant or single cells in tissue culture. The plasmids with the desired inserted genes passes in various paths. The inserted gene expresses its effect. Agrobacterium tumefaciens normally infects only dicot plants, but the one with modified non-virulent plasmids can also cause genetic changes in monocots. Now comes the question, how will we introduce this recombinant DNA in the host? Or how would we introduce the vectors containing the recombinant DNA in the host? Competence is basically the ability to pick up genes and plasmids from outside. DNA is hydrophilic molecule. It cannot pass through the cell membranes. However, competence can develop for a short duration toward the end of the growth period. Competence can be enhanced by treating the host cell with a specific concentration of divalent cation like calcium. It stimulates the attachment of DNA fragments or vectors over the receptor sites of cell wall. The cells are then incubated on ice followed by a brief heat shock at 42 degrees Celsius and again put back on ice. This makes the foreign DNA pass through plasma and enter the cell. Though nowadays other methods like the ballistic gun, the microinjection and the use of disarmed pathogen is also used for introducing the recombinant DNA into the host.